Okay, here is a set of human chromosomes, and this is taken from a Wikipedia page, and it's called a karyotype. So to make a karyotype, a person's cells which undergo mitosis, of course, is the only time the chromosomes are visible, are photographed, and then they're ordered like this. And you can see in a human being, there are 23 homologous pairs. Okay, so starting off with, they're numbered from in order of size, starting off at number 1. And if you see down here, we have got these are the sex chromosomes, X and Y. And this shows that this individual must be a male rather than a female. And it's a bit unusual. If you look at in the case of the X chromosome, a female would have 2 X chromosomes, and they're the same size of each other. A male has 1 X chromosome and a much shorter Y chromosome. Right, so that's a normal human karyotype. If changes occur to the chromosomes, that can result in disease states, and it's called chromosomal mutation. So probably the best known chromosomal abnormality is when you get 3 copies in a person of chromosome 21. Of course, you should have 2 copies. So if you look, there's chromosome 21. It's a one of the smallest chromosomes. In Down syndrome, a person suffering from Down syndrome, you'll see that there are 3 copies of chromosome 21 instead of 2 of them, and it's sometimes called trisomy. And it has fairly profound effects on the individual. There's no mutation to the DNA here, but you've just got too much of one particular chromosome. It appears that the dose of a gene is also important, as well as the actual gene itself. So you're getting too much of it. It has really big effects on a multicellular organism, and it would appear that the effects are most important during the development of the embryo. And as is well known with Down syndrome, it has multiple effects on the person who suffers from it, multiple health problems. Right, how does Down syndrome occur? Well, it occurs from non-disjunction of the chromosomes during meiosis division 1. So if we just look at what happens there. So I draw a cell. Sorry, I draw a cell. Let's say this is a metaphase, so that's of meiosis 1. So I'll just draw 2 pairs of chromosomes, 2 homologous pairs. So there's one big one, and we'll say there's another small one there. Obviously, I'm not going to draw all 23, but we'll get the idea of what happens. So here is the cell at metaphase. The chromosomes have lined up on the equator of the cell. The spindle will be attached, and what should happen is, of course, that you're going to end up with one daughter cell, or one cell which has, it's got the haploid number of chromosomes, so it's got 1 of each homologous pair. And that, okay, so those go on to meiosis 2 to form the gametes. So you can see in trisomy, what will happen is both of these chromosomes, instead of one going each way, one that way and one that way, let's say they're both going to go this way. So you'll end up with 2 copies of chromosome 21 there. Let's say 21 is the bigger one. And of course, these should be haploid. It's got too many chromosomes. So when this goes on to fertilization, the resulting individual is going to end up with 3 copies of chromosome 21. Right, so you may think, right, so if that is the case, actually, do we ever see this gamete here would have no chromosome 21. So do you ever see individuals with, instead of 2 pairs of chromosome 21, with just one? Because one of their gametes, so one of their gametes is this one here. It's only got 1 copy of chromosome 21, so the individual would only have 1. You'd never see that in a person, probably because it's incompatible with life. So that embryo, when it develops, it just wouldn't get past the early stage, and it would fail to develop. Right. So let's think about: do we ever see trisomy with other chromosomes? It's much rarer. Chromosome 21, which is Down syndrome, is by far and away the most common one. You do see trisomy with occasionally with chromosome 18 and also with chromosome 13, very rarely. With these, the infant, the only 10% of them survive to the first birth date. So the effects with trisomy of those is a lot more severe than with 21, and the problems, the medical problems, are much greater. You don't see trisomy with any of the other chromosomes, presumably because if that does occur, then that embryo isn't compatible with life, and it will die. These non-disjunction events that cause trisomy seem to be more common in Down syndrome. It's usually it's non-disjunction in the mother, so in the formation of the female gamete rather than the male gamete. And you probably know that the chance of Down syndrome occurring increases dramatically as a woman gets older. It's a sort of exponential increase as the woman gets older. These non-disjunction events seem to occur more frequently. There are some non-disjunction trisomies of the sex chromosomes. So occasionally individuals will have X, X, Y. They'll be outwardly male, but they've got they're usually infertile, and they have other medical problems as well. Now, this must occur from non-disjunction in the male sperm, because if you think about it, well, when you form during meiosis, you should have in the male you have a cell with 2 X chromosomes, undergoes the first round of meiosis, and you should end up with one with an X and one with a Y. If this one meets up with a female egg, then you're going to get an XY individual. And if this one meets up with the egg, you're going to get an XX individual, a female. If you get a non-disjunction event, and so that, you know, one of them ends up with no Y chromosome there, and you get an extra Y chromosome there, if this joins a fertilization, that's going to give you XXY as the individual. Okay, so these are abnormalities of the karyotype, which are present in every cell of the body, and they result from something faulty going on in meiosis. Abnormal karyotypes are seen in, if you look at tumor cells. Okay, so tumor cells often have, they will not have this, the correct number here at all. They quite often have duplication of number of chromosomes, deletions of chromosomes, bits of chromosome missing, and these chromosomal changes can be the cause of a cell becoming tumorous. In a later module, you may want to look at, there's a later video on it, but if you get a duplication of a chromosome, so you get more copies than you should have, and it contains an oncogene. Okay, so Oncogenes are genes which can make a cell become cancerous if they're overexpressed. And if you've got more copies of that oncogene than you should have, say if you've got 3 chromosomes instead of just 2, then that may be enough to tip a cell into becoming cancerous. Also, if you get a deletion of a chromosome and it contains a so-called a tumor suppressor gene, tumor suppressor genes are genes which are involved in controlling cell division. So if you get a deletion and you lose one of these tumor suppressor genes, again, then you lose the control over cell division. That may be enough to make a cell become cancerous. So chromosomal mutations do cause disease. It's not, you're not getting changes to the DNA there, but you're getting changes, as we said, to the, if you like, the dose of gene. You get too much of the gene or not enough of it, and that is very important. And that's particularly important in early embryological development. So if these problems with the chromosomes occur at meiosis, then the embryo is not going to develop properly. If it occurs later on in adult life, then it may just cause the cells to die, or as we said here, it may be enough to make a cell become cancerous.